Okay, welcome to our first lecture. Well, it's the first lecture I'm doing. I'll catch up with the other lectures from the first week later on, but I think it's important for me to get these second week lectures out to you for Friday so you can study up on them. So here is the first lecture I'm doing for conservation biology, and that is, what is biodiversity? In the first week we talked about what is conservation biology, or the conservation of biodiversity. This week we're going to talk more about what biodiversity is and what we're actually trying to conserve. So in this lecture I want to talk about the definition of biodiversity, what biodiversity is, and the different levels of biodiversity. You've probably heard this: the genetic level of biodiversity, the species level of biodiversity, and finally the ecosystem or biome levels of biodiversity. And then I want to talk about patterns of species richness in the different parts of the world, and what biodiversity has been found in the different taxa. And finally I'll take a look at some of the futures of biodiversity studies. So first of all, what is biodiversity? Let's have a definition of biodiversity from the book. Biodiversity is the variety of living organisms considered at all levels of organization, including the genetic, species, and higher taxonomic levels, and the variety of habitats and ecosystem, as well as the processes occurring therein. So basically what biodiversity is all living things, how they are put together and how they interact with their environment to form ecosystems and so on. Now this definition I think might have come out of your textbook, Primac, but it's generally a similar sort of situation in most textbooks and maybe even in Wikipedia. I don't know. You should probably go and take a look. I want to talk about, first of all, that first part of that definition, the all levels of organization, including the genetic, species, and higher taxonomic levels. So let's first take a look at the different types of biodiversity, the different levels. First we have genetic diversity. So genetic diversity is the diversity within a species, okay, within a unit which breeds with itself. Okay, so genetic diversity usually expresses itself to the naked eye as different phenotypes or different configurations of features on different individuals of the same species of plant or animal. So, for instance, genetic diversity within the human race is expressed by differences in skin color, hair color, eye color, height, propensity to obesity, and different things like that. So genetic diversity is the first level of biodiversity that we are going to talk about. The second level of biodiversity that we can talk about is species level diversity. So not only do we have differences within a species, but within a particular area we may have lots of different types of species. So in this marine ecosystem example, we've got many different types of fish, coelenterolids, seahorses, and squid. So we've got many different types of species within this particular area, and that is known as species diversity. And generally when people talk about biodiversity, quite often they are really talking about species diversity, but things like genetic diversity are also equally important. Another level of biodiversity is the community or ecosystem level biodiversity, and in this example it's represented by different habitats, like this coral habitat here, a sponge habitat over here. Okay, so we could maybe a hard bottom habitat and a soft bottom habitat. Excuse me. So these two ecosystems, different ecosystems occur in the same area, and they make up community or ecosystem diversity, which consists of many different species. So as you can see, we have different levels of biodiversity, different organizations of biodiversity. Now, the reason why we consider biodiversity on these different levels is because different problems of conserving this biodiversity may require solutions at different levels of biodiversity. So let me explain what I mean by that. If you are, say for instance, a cocoa grower, okay, or a scientist dealing with cocoa, the level of biodiversity which would be important to you would be genetic level biodiversity. You're interested in the species cocoa, but you want different varieties of cocoa which are going to give you different things like resistance to diseases, different flavors, abilities to grow in different types of environments. So if your problem is cocoa and trying to maximize productivity, or an agroecosystem concerning cocoa, you would be concerned with conserving genetic biodiversity, and specifically genetic biodiversity of cocoa. If, on the other hand, you are a member of the forestry division who has been given the mandate to help conserve game animals, so animals like agouti, deer, lap, tattoo in the forests of Trinidad and Tobago in particular, then you would be concerned with conserving at a species level. You want to maximize the number of species that you conserve, of these game animals, that you can conserve. And because of that, you would probably be interested in species level biodiversity. So if you want to set up a protected area to conserve as many species as possible, you would be looking at maximizing the species in your protected area. So you might choose an area where all of the game animals are present, rather than choosing an area where there's just one of these game animals present. Okay? Good. So let's move on now to the first level of biodiversity, genetic biodiversity. Now, this is the ultimate source of biodiversity at all levels. It really is a representation of a library of different molecules which code for proteins to be expressed in different ways. And the numbers of genes which make up this genetic diversity is over more than ranges over more than 3 orders of magnitude, and there are always more combinations in ways in which these genes can be put together than there are individuals of a species. So there is never a point where one individual is exactly identical, has all the same genes as another individual, because that is just very, very unlikely. The chance of all those genes occurring the same, unless it's because of a twin or some sort of accident at reproduction, the chances of genetically identical individuals from different parents and so on is exceedingly remote. So each individual is genetically different. So we've got an example here of snails. Lots of different patterns on their shells from different individuals. So within a species, the possible number of combinations of genes exceeds the numbers of individuals within that species present. So there is always more combinations than there are individuals. That means there is always going to be, every individual is going to be unique, unless, of course, as I said, one is the clone or there's some sort of twinning upon reproduction and so on. Measurements of genetic diversity have many theoretical and practical applications. One of the practical applications I outlined was with our cocoa researcher. But if we measure genetic diversity, we can get an idea of whether a species needs work to be conserved or not. If the genetic diversity tends to be low, that means that that species is likely not to be able to respond to changes in its environment. Okay? Certainly not as well as a species which tends to have a much higher genetic diversity. And the reason for that is genetic diversity tells you that there are individuals within that population which are doing things all in a slightly different way. So if the environment changes, there is more likely to be an individual within the population which can cope with that environmental change or even benefit from that environmental change. Okay? If the genetic diversity is too low, it's much less likely that an individual will occur which can benefit or survive that environmental change. So therefore a species with low genetic diversity will tend to be less stable or more likely to go extinct than a species which has higher levels of genetic diversity. Okay, so some of the ways in which we can structure genetic diversity. Genetic diversity is the basis of it all is within the individual, the two different genes within the two different alleles on the same gene of a particular individual. That's the first level of genetic diversity. So here, the gene A is more diverse than the gene B within that individual. Okay? So you can see A is different to little a, but in gene B, big B is the same as big B. So big B allele is the same as big B allele. So genetic diversity starts right at the chromosome level. So chromosomes can also be very different or diverse or less diverse. If you have chromosomes, one from each parent, you tend to have a greater diversity than if the two chromosomes come from one parent. Okay, maybe clonal. Okay? That variation will occur within an individual. Within a population, genetic variation may also be high or low, as I mentioned before. If one individual is pretty much the same genetically to all the other individuals, genetic variation is low. But if one individual is very different to all the other individuals, then genetic variation may be fairly high, certainly within a population. Now, a population is a term which denotes the individuals of a species which breed together. Okay, and there may be several different populations. So in this example, there will be a population of these bighorn sheep on one mountain, and they would be isolated from the population of bighorn sheep on a different mountain, which will be isolated from the different mountain again. Okay, so if this population is genetically distinct from this population, we're talking about genetic variation among populations. Okay, if this population is more or less has the same genes in the same proportions as this population, then the genetic diversity is low among populations. And what that probably means, if the genetic diversity is very different among populations, those populations have been isolated for a long period of time. If they're the same, then these populations are probably not too isolated. Okay, so we can also tell what's happening with those populations by looking at their genetic material. Now, genetic diversity varies very much with the size of the population. As a rule of thumb, genetic diversity, in this case termed heterozygosity, tends to be much greater in a larger population. Okay, and in many animals, that is almost a linear relationship. So generally, the larger the population, the greater the genetic diversity within that population. Now, why that is, you're going to find out later on in the course when Dr. Austin comes in and lectures to you about population sizes and genetic diversity. Okay? Right, so genetic diversity, or what we're calling it here, intraspecific variation, which is the same as genetic diversity. If it is high within a population, the potential rate of evolutionary change is proportional to the genetic variability. So, if you have high genetic variability, the potential that you have for evolving and changing to meet environmental changes tends to be high. If you have low genetic variability, you are much less likely to be able to evolve, and in particular to cope with environmental change. Levels of genetic diversity within between populations, if it's high, then this is resulted from local adaptations, but it also means that these two populations are more or less isolated. Now, these local adaptations are always going to build up. If a population is isolated from another population, some genes may be lost, as an individual which only carries those genes is lost from the population before it breeds, for instance, or if mutations occur in that particular population and those mutations are conserved within the population, in other words, they're not negative mutations, then that mutation will only occur in one population. Okay? And these things will build up genetic diversity between populations. And Dr. Austin will talk more about that later. So there's two different ways in which genetic diversity can build up. Okay? If you have different genotypes within the same environment, they are going to express as different phenotypes. If you have the same genotype in different environments, then they are almost always going to be selected differently and eventually result in different types of species or different phenotypes of the same species. Okay? Because the different environment is going to exert different selective pressures on the same genotype. Okay? So when you see different phenotypes within a population, you need to know it's not necessarily meaning that there are different genotypes. It can mean that different environments are working on the same genotype to give you different phenotypes. Eventually, that's probably going to mean that these are going to be different varieties. Okay? So, if we look at evolution, these are the Hawaiian honey creepers. Okay? Their ancestor was a single species of some undifferentiated sparrow-like bird, and with time its descendants diversified into these many different types of species. Okay? And that was through the inherent genetic variability that it brought with it. Excuse me, I will just deal with that. Okay, sorry about that. So we were looking at Hawaiian honey creepers, how the inherent genetic variation in the first individual would help to have given rise to these many different species, but also mutations which occurred while this species was on the island as well. So these two different processes would give a rise to this great range of different species. Okay? Alright, let's move on now to the next level of biodiversity, and that is species level biodiversity. Generally, when we talk about biodiversity, most people think of species biodiversity. So we can think of species biodiversity or represent it or measure it in several different ways. Quite often when we're planning conservation effort, we usually think of species level biodiversity. What is the maximum number of species that we can save in a protected area or with some particular amount of conservation effort? So where we direct our conservation effort, for instance establishing a protected area, we may want to maximize, quite often we want to maximize the number of species that we are going to conserve. So it's important that we understand the different ways in which we can represent species and measure species level biodiversity. The most simple way of measuring species level biodiversity is by just counting the number of species. Okay? And this would be species richness. This is called species richness. Now, some people would just be working on one particular taxa. So for instance, I would work on plants, other people would work on birds or butterflies and so on. The numbers of species in these different taxa is not necessarily independent. So if you have a greater species of plants, for instance, quite likely that would mean a greater species of birds, greater species of butterflies, for instance. If you have a greater species of birds, it may indicate a greater species of tree and so on. So that gives rise to the concept of indicator species. So you may choose a particular species like birds or butterflies and use that to estimate the species richness in an area. Okay? Species richness in an area of all the taxa. So if you have an area which has a high species richness of birds, it's quite likely to have a high species richness of plants as well. And that can be used on a much larger scale as well to identify parts of the world where high levels of species richness occur, or the hotspots of biodiversity. We can also use a measure of species diversity called species diversity indexes. Now these are an advance on species richness by also looking at the evenness that the individuals of a particular taxa are spread between the species. So the idea being that if you have two areas which have the same species richness, in other words, the same number of species, you can distinguish between them by looking at the numbers of individuals within each one of those species. If there is a large number of individuals within each one of those species, then those species are much more likely, first of all, to survive and persist in your conservation protected area or whatever. And also no one species is going to dominate the ecosystem. So we would tend to take the area which has a greater evenness of spread of individuals within the species or between the species. Okay? So there are two commonly used indices, the Shannon index and the Simpson index. Okay? They both measure not only the number of species, but they also incorporate the proportions of individuals between the species. Okay? You may use numbers of individuals or abundance or biomass or productivity of the different species. Now, these indexes are useful, but because not everybody carries them out, they don't tend to be used as much as, say, species richness. Species richness is very much easier to describe because you don't have to count any sort of abundance or biomass. There's no measure of abundance in your sample. All you have to do is count species. Okay? So these species diversity indexes, which convey more information, may not be used as much as, say, species richness, which conveys much lower amounts of information. Okay, so what are the levels of species diversity? First of all, let's talk about the levels of species diversity on a global scale. There's approximately 1.5 million living species described and given scientific names. And there's about 300,000 fossil species which have also been described and given scientific names. Now, does that mean that there are 1.5 million living species in the world? No. It is actually estimated that there are 10 to 50 million species of plants and animals in the world. Plants, animals, and microbes in the world. The reason why I've got this figure of 1.5 million is because these are the ones which have been extracted from the wild, closely inspected, had their details described, and have been given a scientific name. Okay? So it's estimated that about 10%, 10 to 20% of all living things have gone through this process, but there is still 90% of living species which still do not have a name, have not been described, and therefore the human race simply does not know about them. And that's a shame, because with the rate of habitat loss and extinction which is going on in the world today, a large fraction of these living species will disappear, become extinct, even before we know about them, even before they've been named. It's a classic example of a ridge in the western Amazon basin. Now, the western Amazon basin is the most diverse area when it comes to flowering plants. Okay, there's more flowering plants per hectare in the western Amazon basin against the foothills of the Andes than there is anywhere else in the world. Now, once a botanic gardens, who will do a lot of collecting of flowering plants and so on, collected in this area, and they went to one particular ridge and they collected extensively up and down that ridge, and they took all the specimens back to the States. Over the period of the next 5 years, they worked through the specimens, they identified them, they described all the new species and gave them scientific names and so on. And they found a very high diversity. So they wrote back to their collaborators in, I think it was Ecuador or Peru, and they said, you really got to conserve this area because it has such a high species diversity. Okay? And unfortunately their colleagues in Ecuador wrote back to them and said, I'm afraid that ridge that you collected on has been cleared for cattle farming. The plants and the ecosystems on it no longer exist. Okay? And many of those species were only found on that ridge. So it was only by accident that they were actually known about before they were made extinct. So that's a sad story, but unfortunately replicated only too much around the world. So what are the distributions of these species between different taxa? So what is the most common type of life on Earth? Okay, when we look at the species which have been described, we get a pattern such as this. Okay? By far the largest number of species which have been described and scientifically documented are the insects. Over 750,000 species of insect have been described. Okay? The next largest number of species are the plants, with about 250,000 species of plants. Okay? And I want you to note the number of species of viruses which have been described. It's only 1,000. Okay? Number of species of fungi, only about 70,000. Okay? So these are the number of species which have been described and which make up that 1.5 million that we mentioned earlier. Now, does this mean that these are all the species which are on Earth? No, it doesn't. And in fact, there are some groups which it is thought have barely been studied or barely been described and examined. In particular, the viruses, the bacteria, the protists, and the unicellular algae. Also the nematodes, mites, and also still the insects. It's thought that these groups are there are millions of species in them which simply have not been described. So even though they haven't been described, they're still there, and they make up the rich diversity of life on this planet. So this graph gives you an estimate of how many species it is thought exist in the different taxa. Okay, so down here we have the blue portion of the bar shows you how many species have actually been described, and then the green extension on the bar indicates how many species it is thought that exist, actually exist. So it is thought that the majority of plants have been described. The majority of vertebrates have been described. Okay? There's only a few fish and a few lost mammals, maybe a couple of bats, which have not been described. So most of the vertebrates tend to have been described. The same token, plants, they don't run away, so they're easy to collect. So a large proportion of the plants have also been described. Maybe as not as much as the vertebrates, but certainly a large proportion. Insects, the largest number of species which have actually been described are the insects, but it is also thought that there is a huge number of insects still to be described. Insects are an incredibly successful group when it comes to species level biodiversity. Okay? Now, the groups which have not been described very well at all. One group which is thought to be very speciose, has a lot of species, is the fungi. And if you look at the small number of species which have been described in the fungi, there are many more left to find. One of the reasons for this is that these fungi, along with bacteria, a lot of them occur in soils, and they are very difficult to identify from soils. Bacteria also suffer from a nomenclature system which recently broke down under the strain of all the new species of bacteria which were being described. And that nomenclature system had to be completely revised. Same with the viruses. And so bacteria and viruses are both in the process of being described once again. The pattern is also the biodiversity which humans can see and readily sample are the ones which are most well known. So biodiversity or species which are microscopic or cryptic, like the crustaceans, hide under rocks everywhere and so on, or algae which tend not to be important to humans. They just aren't described. So they're very poorly described. So there are many millions of species, particularly insects, which are still to be described in the world today. All right, let's move on now to higher level taxa diversity. So we'd have genetic level diversity, which equates to varieties, and we would have species level diversity. We can go one up in the taxonomic hierarchy into kingdoms, phyla, and classes, okay? And we can look at the diversity amongst these higher level taxa. Now, these higher level taxa are going to be more genetically distinct from one another because they are most likely to have been separated for much longer than species level divisions and genus level divisions. Okay, so they have been reproductively isolated or isolated from one another for much longer periods. So they are much more distinct. They quite often have very different body plans. Okay, most vertebrates have 2 arms and 2 legs. Those 2 arms may be transformed into wings or whatever, but that body plan is still basically the same. If we compare our phyla to the insect phyla, where the insects have 6 appendages, 6 legs, and maybe even a couple of wings. Okay, so that's a very different body plan, indicating a very different type of organism, very different type of genetic makeup. Okay, so a greater amount of separation. Now, because most of life on Earth, or if not all life on Earth, originated in the oceans, okay, it's no surprise then that the organisms, the higher level diversity tends to be found mainly in the oceans because they have been separated right at the beginning, when there was no life on land, there was only life in the oceans. Okay? So these organisms would have separated way back then. So the phyla would have arisen way back then. And it's only afterwards that a few, or maybe half of those phyla actually made the transition onto land. Okay? So, of the higher level diversity, higher level taxa, there are 35 phyla of metazoans or multicellular animals. 34 of those are marine, 16 over half of them, no, about half of them, are only marine, so they're only found in the oceans. Some of them, or about half of them, have made the transition onto the land, but only a few are exclusively terrestrial. Only a few have been separated long enough to be considered completely different phyla. Okay? So therefore we can say that the genetic variation in the marine environment is much higher, okay, than compared to the terrestrial environment. Terrestrial environment may be much more productive, okay, it has a greater rate of productivity, more energy fixed, more biomass and so on. However, it's in the marine environment where genetic diversity is highest, because that is the cradle of all living things on Earth. So the evolutionary potential is greatest in the marine environment. Okay? I'm not too sure why I have this slide here. Maybe it's just to show the great variety of different phyla that you find in the marine environment. This picture shows a weird and wonderful organism which was recently found living on the feelers of a lobster crustacean or shrimp off Norway, and it was found that this organism belonged to no phyla known. So a new phyla was erected for this organism. Okay? So, particularly in the marine environment, we may find even new phyla. Okay? I have no idea what it's called. You can take a look at the details of the picture in your textbook and take a look and see what it's called. Okay, moving away from taxa, we can also express biodiversity in terms of ecosystem and biome diversity. So what is an ecosystem? As you already know, it's the biotic community and its abiotic environment functioning as a system. So for instance, a freshwater pond, or a grassland, or a forest. Okay? So there's usually boundaries between them, but one of the things which militates or makes it difficult to assess between ecosystems is those boundaries can tend to be very blurred or gradual. Okay? Similarly, biomes, the boundaries between biomes, may also be gradual. Okay? Biomes are major regional ecological communities. In other words, collections of ecosystems which have a similar characteristic. Okay? So a tropical rainforest biome has many different types of tropical rainforest, maybe with different degrees of deciduousness or something, or different species. Okay? So in an area, if an area has a high ecosystem level biodiversity, then it has many different ecosystems. Trinidad is a good example of an area, a country which has high ecosystem level biodiversity. We have many different ecosystems, all within a fairly small area around Trinidad, and that's a function of its position. It is an island, It is an island at the boundary between rainforest and maybe dry forest biomes. It has marine environments, it has coastal environments, and it has terrestrial environments. So it has high levels of ecosystem biodiversity. Okay, and we revisit this picture to demonstrate ecosystem biodiversity. We have soft bottoms, we have hard bottoms. Quite often we'd have different organisms in the different ecosystems, but some of those organisms are going to be the same, so these sponges occur in both ecosystems, for instance, and you may find that the animals flit from one ecosystem to the next. So the boundary between these two ecosystems can be blurred. The biomes, this is probably the biggest level of biodiversity. There are some parts of the world where 3 or more biomes intersect, and they would have high biome diversity. I don't think there's more than 3 in any small area. So we'd have different biomes. So this light green is the tropical rainforest biome, which occurs around the equator. Okay, the orange biomes are the desert biomes, and the light green here is, or the khaki green here, is the temperate deciduous forest biomes. Dark green is the boreal forest, those conifer forests, and this would be the tundra biome. Okay? Ecosystems and biomes can be difficult to classify because of the boundaries between them being gradients, but they generally tend to be classified based on their dominant plant communities. Whether or not they are dominated by trees makes them forests or not, grasslands or not, shrublands or conifer forests, and so on. Marine ecosystems are even more difficult to classify, but we would have things like seagrass meadows, coral reefs, sandbanks, and so on. Alright, let's move back to species level diversity now, and I want to take a look at species level diversity through geological time. Has species level diversity, and we'll use that as a proxy for biodiversity, has it remained the same throughout geological time? Has there been the same number of species from the beginning of life on Earth, and has it remained fairly constant throughout the 4 billion or so years that life has been on Earth? Okay? Let's take a look at that. And the answer to that is no. Species richness, and we assume biodiversity, has gradually increased over geological time. Okay, we can tell this from the fossil record. We see a greater and greater diversity of different taxa as the geological time period goes on. So here's an example. These are the terrestrial plant species. Okay, the sum of all terrestrial plant species is the top line, and it goes up and up from the Silurian all the way up to the Tertiary, 420 million years ago down to 2 million years ago. Okay, and this is based on from the fossil record. Now, as you can see, they've separated these fossils into 3 different groups: the pteridophytes, or the ferns, the gymnosperms, or the conifers, and then the flowering plants. At different periods, these different types of taxa have dominated, but they've all contributed to a gradual increase, okay, in the number of species throughout the fossil record, and therefore throughout geological time. And this type of increase over time is reflected in pretty much all the other taxa that we have a good fossil record for. There have been fluctuations in this slow, steady increase in the number of species, where the numbers of species disappearing has outstripped the numbers of species appearing. So, for instance, during the Triassic-Jurassic boundary here, in the transition between the Triassic and the Jurassic, there was a steep dip in the number of species of gymnosperms and pteridophytes. Angiosperms hadn't evolved yet. Okay? So the total number of species decreased quite sharply at this boundary. Okay? I think this is the boundary where it's speculated that a large meteorite hit the Earth and caused the extinction of the dinosaurs. Okay? So we would have decreases. We may also have decreases in particular taxa. So for instance, the pteridophytes, the ferns, decreased in the number of species. Okay? And this was mainly because they began to be outcompeted by the gymnosperms over time, and the gymnosperms' number of species increased at the expense of the pteridophytes. When the flowering plants came on the scene, they likewise decreased the number of gymnosperms and took more of the resources for themselves. But these dips in the numbers of species occurs at different points in the fossil record in geological time. And overall, the species richness of most taxa has increased with time. So we're living in the most biodiverse time that the world has ever seen. Unfortunately, we are in the middle of one of those mass extinction events. So we are losing that peak of species. So what do we need to do? Well, one of the things which most scientists agree on is that we must understand where all that biodiversity is. If we don't know where it is and how much there is, then we don't have much chance of being able to save it. So some biologists recommended initiating an intense global biological inventory aimed at discovering and classifying all species. The amount of resources that would require would be huge. The human race could probably do it. I think we've got the resources, but the political will and the people wouldn't want to pay for it. Others recommend the rapid recognition and preservation of threatened habitats that contain the largest number of endemic species. This is a more practical approach because it recognizes the fact that we are not going to be able to save everything, and we are not going to be able to save everywhere. So let's find the areas where there is the greatest number of species and conserve them. This also requires us to study biodiversity, to find out where that biodiversity is. So really, the future of biodiversity studies is very much, it's got a long way to go in terms of describing. Okay? So these are some pictures which indicate some of the new and innovative methods which are being used to try and describe biodiversity in the areas where it's least described. And hopefully I got a lecture there which tells where biodiversity is least described, and most effort is being put in to finding that biodiversity. This is in French Guiana in the rainforest there, sampling insects from the forest canopy. This is in Costa Rica or Panama, where they're spraying insecticide into the canopy to kill the insects so they'll fall down and they can collect them. And this is the end result. Thousands of hours of painstaking sorting and separating and taxonomic work to describe all the new species which are found. And as we saw earlier on in the lecture, the insects have got a long way to go. And there are also other strategies suggested by different workers. E.O. Wilson is a very famous ecologist specializing in ants and other arthropods. He suggests rapid assessment programs, a team of scientists specializing in different taxa going into an area and inventorying as much of that area as possible. If you've all heard of the BioBlitz approach, that the rapid assessment program spreads a BioBlitz effort over the course of a couple of months rather than a 24-hour period. There are other approaches, the biotrop approach and the monographic studies of different organisms. But it's generally agreed that we need a lot more effort in describing and finding out where all these different species are. And that is one of the frontiers of biodiversity. So if you want a job as a taxonomist for spiders or nematodes, you will be definitely well needed in different parts of the world as these organisms are gradually explored and described. Okay, well, that's about it for this lecture. Thank you very much for watching.